So many of you who know my work will know of what I've written about biopolitics and neuropolitics. But actually I'm going to talk about something a little bit different today, although I hope you'll see that when we get to the end of my talk, these ways of arguing come together. But that's for the end. So what I'm going to talk about today is whether or not recent developments in the life sciences and biomedicine have any implications for the human sciences and in particular for sociology. And I suppose I'm going to end up with talking very briefly about some work from a French anthropologist called Didier Fassin. At one point in his work, Fassin says, perhaps it's time to move from analysing the rules of the game to engaging with the stakes of the game. So this is a little attempt to engage with the stakes of the game. So what difference does it make that humans are living beings, that they're animals? What difference does that fact of the living nature of human beings make to the social and human sciences? Because humans are, after all, primates. They're very special, they're distinct, but they've got affinities with other living creatures who are born, who live, who reproduce, and ultimately who die. And if that fact, that human beings are living creatures, if that fact makes a difference, then how should we in the social and human sciences respond to that? How should we respond in particular to the waves of new knowledge that are coming towards us from the life sciences, from genetics, from biomedicine, and from neuroscience? Now, I've been working for the last 10 or 15 years amongst life scientists and for the last 7 years amongst neuroscientists. And in my work with those researchers, I've come to the conclusion, not me alone, but many others, that human ontology, what we think we are as human beings, is being remade by the life sciences, by genetics, by neuroscience, and by biomedicine. And in the course of thinking about that, I've come to the conclusion that we need to seize this moment to rethink the relationship between the human sciences and the life sciences. I don't think when we rethink this relationship we should engage in naive enthusiasm. There are many in the human sciences who are rushing towards neuroscience, embracing it fully and believing they can remake their own disciplines on the basis of neuroscience. And I think that's naive. And there are many also who've grasped what they take to be the opportunities of epigenetics, which I'll say a little bit about later on, to think that now at last we can understand the way in which the genetics and the social are linked together. Again, I think we need to keep our critical wits around us. We shouldn't engage with naive enthusiasm, but nor do I think we should retreat to what has been the traditional position, at least over the last 20 or 30 years, the traditional relationship between the social sciences and biology in particular, which is to treat any reference to the biological in human affairs with suspicion and with critique. The relationship I would like to propose, and it's a relationship that I talked about when I wrote that book with Joël Abi-Rached, Neuro, is what we called a relationship of critical friendship with the life sciences. And what I want to do in this little talk today is to try and illustrate what that critical friendship might look like. But first, why do I think it's a transformative moment in the relationship between the social sciences and the life sciences? We know from our history that these two disciplines, biology and sociology, were born very close together in the 19th century. I've just put on the board here, but you can look that up for yourself from the Oxford English Dictionary online, the timeline that they set out for the emergence of these two disciplines. Biology, as we know well, was born in 1802, and with the birth of biology, nature was divided into two kingdoms, those that were possessed of life and those without it. 1802. Sociology, well, the origin of the term is a little bit disputed, probably first used in 1780 by Sieyès and then adopted by Comte in 1839, coming slowly to stand for the emerging science of the human moral order. From that time onwards, that is to say from the early 19th century, there was a very close and rather contested relationship between the social sciences and the life sciences. The sciences of the human moral order and social order were repeatedly differentiated from those of other living things. But at the same time, as we know, the social or moral order was repeatedly understood with biological or quasi-biological terms. Metaphors from biology and styles of thought from biology were imported into the social sciences right across the 19th century and into the 20th century. You only have to think of the role of the idea of evolution in the social sciences, or the crucial role that the biological notion of degeneracy played in the social sciences and in the development of eugenics. And not least because of the overriding concern in the 19th and early 20th century with the question of population. Now, I've done some work on the history of British sociology with my colleague Tom Osborne, who's a professor of sociology at Bristol, and explored the way in which the concept of population was absolutely central to the sociology, at least in the UK, in the early 20th century. The laws of growth of population, the laws of fecundity, of morbidity, of mortality, the factors affecting the quality of the population. In fact, if you look at the biographies of many of those who were key in sociology in Britain in the early 20th century, many of them actually started off as biologists. Now, of course, we know that one way of posing this question was in terms of eugenics, but I think it would be misleading to think that all those people who were concerned with population were concerned with eugenics, at least insofar as we understand the term of eugenics as a kind of program to eliminate those pathological individuals, prevent them from breeding, and remove them from the gene pool of the population. Many of those who were concerned with population were concerned with it in a different way. Indeed, the famous British sociologist Richard Titmuss, who wrote a very powerful critique of early forms of marketization, the gift relationship, some of you may know that, was actually secretary of the Eugenics Society quite late in his life. And no one can say that Titmuss was himself a eugenicist in that way that we commonly think of that term now. So this close relationship between the sociological and the biological that one sees across the 19th and the first half of the 20th century probably continued up until around 1940. And of course we know that from 1940 onwards things became rather different in the wake of the Nazi spiritualization of the biological and their biologization of the spiritual. Although the reduction of interest in eugenics was probably slower than you think, especially in the United Kingdom, when the Eugenics Society continued right up through the 1950s and the 1960s. Nonetheless, by the 1970s, it became sociological common sense that fatalism, determinism, reductionism, sexism, all those other undesirable things would follow almost inevitably from any reference to human biology in explaining human social relations or human individual conduct. Whether or not that was posed as a an ontological question, what were humans really like, or as an epistemological question, what can biology tell us about the forms of life that humans have made for themselves? Human biology was relevant, it seemed, only in that it provided the preconditions for language, for meaning, and for culture. But the form and the content of language, meaning, and culture had to be explained in non-biological terms. In the famous words of Clifford Geertz, humans lived in the web of language that they themselves had spun, suspended in webs of significance that they themselves had spun. And of course we know that that wariness about the biological was very well grounded, because one only had to look at some of the immense simplifications that biological arguments and sociobiological arguments undertook about human nature, the claims that that aggression, that sexual difference, etc., were fixed by human biology. One only had to look at those simplifications to believe that biological arguments were inescapably on the side of reaction, and that to be on the side of progress, of human progress, you had, in a sense, to take a stand against the biological. Any reference to the biological was on the side of a kind of reactionary politics that tied human beings to a fixed kind of nature. Of course, that didn't mean that people were not concerned with the body constructed, the body represented, the body performed in language, in culture, the body addressed by the new assisted reproductive technologies, and so on and so forth. There was an intense interest in the body, but somehow that interest in the body evaded a concern with the fleshly, bloody thing itself. So what licenses my claim that this is a potentially transformative moment in the relationship between the social and the human sciences? Of course, we can already see that things have started to change. We've already seen the turn away from representation, the turn towards the body, the turn towards the material in a whole range of areas. We see the rise of a new feminist materialism, illustrated on this slide here by that book, Meeting the Universe Halfway, which many of you may have read. We see the rise of zoontologies, the claim that humans and other animals do indeed share something in common, and that our complacent belief that only humans have language, only humans have meaning, that only human life is a worthy life and animals are there to serve us, and needs a radical rethink. And one sees it also in the rise of a certain kind of theory within progressive social science, affect theory, that draws to some extent on contemporary neuroscience in arguing that the nature of human beings and what mobilizes human beings are forces which operate at the pre-conscious or the non-conscious levels, flows of affect that bind human beings into certain kinds of relationships. But from my point of view, many of those ways of taking up the life sciences do so in a very speculative and philosophical way. And my interest is an interest which perhaps I can most clearly phrase as an interest in the implications of the truth discourses of the life sciences themselves. Not what can we take out of the life sciences and make use of in our own social sciences, but what are the life sciences themselves telling us about the nature of human beings? I think if we look at what the life sciences are telling us about their beliefs about the nature of human beings, we see that the arguments in biology have rather profoundly changed. No longer is the biological considered as a fixed, true nature of human beings. No longer is biology considered destiny. No longer is our fate written in our genes. But something else interesting is happening. And I argued this in that book, The Politics of Life Itself, and also to some extent in Neuro. In our age of biology, the biological is in many different ways, I think, opening itself up to the social, to history, to culture, and therefore to the kinds of explanations that make sense within the social and human sciences. That's perhaps a rather paradoxical claim, because I've argued elsewhere that in the so-called age of biology, with the rise of what I've called a molecular vision of life, life is no longer conceived as mystery, but as mechanism. An age of biological control has emerged, in which biology has moved into something which can be engineered and re-engineered. In a peculiar way, however, this idea that biology is no longer mystery but mechanism, a mechanism that can be understood and engineered and re-engineered, is a much more hopeful vision of the biological. In a peculiar way, the more biological something is, the more it is open to transformation in this way of thinking. The more biological, the more it can be understood, the more it can be transformed. I'll say a little bit more about that later. Secondly, perhaps the major critical relationship between the social sciences and the biological sciences had to do with genetics, and the belief that somehow genetic explanations framed human capacities in terms of an unalterable inherited nature that simply worked its way out through the individual's life course. But I think we've seen a radical transformation in genetics. In fact, in the shift from genetics to genomics, from the idea that we have genes for particular characteristics, to the idea that our genes and how they are expressed is transformed over a lifetime as a result of the kinds of experiences that humans have from the moments of conception onwards, if not before. We've seen a move from a concern with heredity, that is to say, inherited DNA, to a concern with epigenetics, which for present purposes are considered to be the activation or deactivation of genetic material across a lifetime in response to experience. And I'll say a little bit more about that later on. That argument about epigenetics has one little phrase that it repeats again and again and again. How does experience get under the skin? How does a human experience, or indeed an animal experience, shape and reshape the body at the level of its genes itself, at the molecular level itself? Experience, adversity gets under the skin. And again, I'll say a little bit more about that in a moment. Perhaps the next key shift that one has seen concerns neuroscience. As neuroscience has begun to investigate the nature of neural processes, it's become clear that the brain, the human brain, but also the animal brain, is perhaps the most modulatable organ of all, the organ that is most open to experience, the organ that is transformed minute by minute, second by second, hour by hour, decade by decade, over an individual's life. The brain becomes the repository of everything that the human has experienced over its lifetime. The brain, that is to say, is plastic. And this idea of plasticity of the brain in relation to environmental inputs, again, transforms biology from something that is static and stands in opposition to the environment to something that is in constant transaction with the environment across the course of the lifetime. We also know that it's not simply the nature of the synapses that change across a lifetime, but also that new neurons can be formed up to the maturity of an adult and indeed into old age in some very elegant experiments by Elizabeth Gould. We used to believe that after a child was a few years old, the number of neurons that it had was fixed, and the only way after that was down. Every glass of wine, every cigarette you smoked, every other insult you did to your body could only lead to the death of neurons. But in these very elegant experiments by Elizabeth Gould and her colleagues, she demonstrated quite clearly that neurons were born and developed across the life course and even into adulthood, and that those neurons and the rate of creation of those neurons was itself affected by human experience. Finally, perhaps in this little catalogue of things that have changed in biology, we also know, or we're coming to understand, the extent to which the human organism is not a coherent unity. Investigations of the microbiome have shown that the human organism is infested by hundreds, thousands, millions of different types of organisms, living in gut, living in skin. The human being lives in a kind of symbiosis with a multitude of different organisms that live within the body itself. In all these ways, and I think in many others, the very distinction between the biological and the social is becoming blurred, between the inside and the outside, between what is given to us by nature and what is given to us by nurture. Those terms no longer serve to demarcate a fundamental division in the nature of the human. So perhaps we are entering an age where that reductionism that characterized biology and the life sciences and biological experimentation across about 50 or 60 years has come to the end of its useful life. Reductionism, I use that term not as a term of insult. Reductionism in experimentation has produced fundamental re-understandings of the nature of the human organism. You break the human organism down into the smallest possible parts, and you see the functioning of each of those small parts. And we know a great deal about the functioning of genetic material, we know a great deal about the functioning of neurons, we know a great deal about the functioning of stem cells, of all those tiny little molecular components of the human body. But what we're very bad at is that hope that was always there, that the more you knew about the tiny elements of life, the more you would be able to build up the picture of life as a whole. It's proved immensely difficult in the life sciences to move from this molecular understanding up to an understanding of the complexity and emergence of biological properties at a higher level. And that, I think, is the fundamental challenge in the life sciences themselves, how you move towards an understanding of vital organisms beyond reductionism. And we'll get to that, I hope, a little bit later on. Now, of course, I'm not the only one who's arguing for a new relationship between the social and the biological. Anthropologists among you will probably be familiar with Margaret Locke's arguments about local biology, that biology itself is shaped and reshaped differently in different cultures and in different localities. Some of you may also know the work of Elizabeth Gross, one of the people who's really over about 20 years tried to break down that distinction between the biological and the social. I'll just read this quote from her because I think it illustrates the kind of ethos that I'm trying to urge upon us. She says it's necessary to redress the foreclosure, the denial of the biological forces that press on and produce life, and thus ironically to overturn the repression of materiality in its most complex forms that has dominated the humanities and the social sciences in their exclusive focus on cultural construction at the expense of natural production. Cultural construction at the expense of natural production, to overturn the repression of materiality. So there are many, I think, who are trying to argue in different ways that we need to overturn the repression of the material, and in this case, the biological, in the way in which we address these fundamental human problems of how life itself is shaped and reshaped in the kinds of worlds we live. Other examples, which I won't talk about, some of you may know Elizabeth Wilson's project on gut feminism, again, which tries to overturn that reduction, that repression of the material. Now, I argue, like Elizabeth Wilson, like Elizabeth Gross, and like many others, that the way in which we need to address these questions is to take the biological seriously, to engage directly with the forms of experimentation, with the forms of argumentation, with the truth discourses of the life sciences themselves. And that's the relationship I call critical friendship. To be a critical friend of the life sciences is, I think, to take a distance from some of the ways in which science has been understood in science and technology studies. Perhaps it is indeed to rehabilitate the idea of truth. I think truth in science is very difficult to make. It's very difficult to make things true in science, because to make things true in science, you have to pass the hurdle of the experiment, the phenomenotechnical hurdle, to use the term from Gaston Bachelard, the attempt to conjure up in reality what you have already conjured up in thought. And if you work with life scientists, you will discover that it's very difficult to conjure up in reality through experiment that which you've conjured up in thought. In fact, it's much more difficult to make things true in the life sciences than it is in the social sciences, where you can make things true by elegant prose, rhetoric, by gathering a few friends together, and by the power of your argument. So we need to recognize something about the truth status of the natural sciences themselves. And we need a relationship with those sciences which goes beyond critique. And in going beyond critique, in trying to collaborate with the sciences, we need to take some dangerous paths. But as I say, in doing this, we should not leave our critical faculties behind us. And what I want to do in the rest of this talk is to give 3 examples of the way in which I think this relationship of critical friendship might best work, and then sum up by suggesting that this does have some implications for how we think about biopolitics in our present. That a certain kind of biopolitics, perhaps a different kind of biopolitics, is possible if one follows these trains of argument. So I'm going to give 3 examples, one from genetics and biomarkers, one around epigenetics, and I'm going to focus on the question of living in cities, and one on synthetic biology, where I'm going to talk a little bit about vitalism. Excuse me. So perhaps genetics is the place where there's been the most difficult historical relationship between sociology and biology. And you probably won't need me to remind you of that relationship, that long history of dispute, that recognition that much of genetics was reductionist, determinist, was fatalistic, the bloody history of genetic argument in politics across the late 19th and into the 20th century with the rise of eugenics, and then with the emergence of a new type of genetic investigation associated with the big science of the Human Genome Project, the belief that that attempt to map and sequence the human genome would produce in its wake a new kind of determinism, a new kind of fatalism, and a new eugenics. This argument was actually very common across the human sciences and the social sciences as that Human Genome Project came into existence and as it did its work. The critics felt that there was going to be a new genetics, that there was going to be a new determinism, a new fatalism, that there would be labeling on the basis of genetics, that there would be stigma on the basis of genetics, that genetics would legitimate inequality, and genetics would lead to a fundamental belief that human abilities and the range of human abilities was shaped by the genes that individuals inherited. Now, of course, there was a huge amount to be critical of in those arguments around genetics and the Human Genome Project, especially in the kinds of claims that were made. But the intriguing thing that has occurred is that the science itself has demonstrated that some of those fears were misguided. So I just want to look at some of the claims that were made and explore this in relation to what is perhaps the leading kind of investigation in biomedicine these days around genetics, which is the search for genetic biomarkers of human diseases. If you read some of the statements around the Human Genome Project as it came into existence, you'll have seen claims like this, and this is a quote from Leroy Hood writing in 1992 in a book called The Code of Codes. This is what he says. Once the 100,000 human genes have been identified, he said, it would transform our ways of dealing with disease. The Genome Project in the 21st century will have a profound impact on medicine and diagnosis and therapy. Perhaps the most important area of DNA diagnostics will be the identification of genes that predispose individuals to disease. However, many such diseases, cardiovascular, neurological, autoimmune, are polygenic. They're the result of the action of two or more genes. Human genetic mapping will permit the identification of specific predisposing genes, and DNA diagnostics will facilitate their analysis in many different individuals. Perhaps, he says, in 20 years, he was writing in 1992, 22 years ago, perhaps, he says, in 20 years, it will be possible to take DNA from newborns and analyze 50 or more genes for the allelic forms that can predispose the infant to many common diseases. For each defective gene, there will be a therapeutic regimen that will circumvent the limitations of the defective gene. Preventative medicine should enable most individuals to live a normal, healthy, and intellectually alert life without disease. This is just one example of the huge hopes that were invested in that Human Genome Project. In 20 years' time, he said, all this will have come about. Well, it's now almost 25 years since he wrote that, and little, if anything, of this has come to pass. We know a lot about single gene disorders for rare diseases. There are actually quite a lot of those rare diseases, about 6,000 rare diseases affecting about 7% of the population, so that's not nothing. But most fundamentally, what's happened is our very understanding of the human genome has been transformed by the sciences itself. And this has led to a radical transformation in how we understand the role of genes in human existence. A move from determinism to probabilism, a move from the genetic program approach to an understanding or an attempt to understand complexity, development, and emergence. And this, I think, opens a new kind of relationship, or a possibility of a new kind of relationship between the social sciences and the life sciences. You'll know the way in which genetic determinism worked its way out across the early 20th century, as I've already said, linked to ideas of fatalism, of fixed hierarchies of disorders, and of dreams of the elimination or exclusion of the unfit or the less fit. And you'll also remember the kind of dreams that there were before the Human Genome Project came to its end. The belief that there would be something like 100,000 human genes, that they would be, to quote, the digital instructions for making a human being, and that you would search for these individual genes and you would use them for diagnostic purposes. And for those who like the sort of media representation of that, this was probably best represented, those dreams or nightmares best represented in that film Gattaca. But as I say, scientific arguments have to face the test of the real, and when the human genome was sequenced, something rather strange occurred, which I'm sure many of you will be familiar with, which is that this idea that human beings, being the most complex of organisms, had the most genes would prove to be untrue. When the human genome was sequenced, it became clear that actually humans did not have more genes than any other, more coding sequences than any other organism, as shown in this little slide here. A fruit fly's got 13,600 coding sequences. That's a planarian worm, has 19,500. Humans have about 20,000. Rice has 45,000 coding sequences. and the humble maize has around 50,000 coding sequences. What that meant was that the very nature of what we thought was going on in genomics had to be transformed. Variations between human individuals were no longer variations at the level of the coding sequences themselves. They were based on small, tiny variations in the chain of DNA that made up those coding sequences, the so-called single nucleotide polymorphisms, variations within a single coding sequence that accounted for the small variations between human populations. And that was the new focus of medical genomics, to try and identify what those small SNPs, single nucleotide polymorphisms, what those small variations were and how they contributed to human disease. And what became evident as a result of those kinds of investigations was that there was no such thing as a normal human genome, that all human genomes varied, and they varied in enormously complex ways that we didn't quite understand, and that some of those genes produced susceptibilities for disorders, and that some of those sequences were protective against disorders. We moved from the idea of genetic determinism to a kind of probabilism, from an idea of the gene for to the idea that there were multiple variations which shaped the probability of getting a disease or resisting a disease, especially when it came to common complex disorders, and even more when it came to psychiatric disorders like depression. It wasn't that there were no politics around these new ways of thinking about human genomics. But what one saw here was a new biopolitics, not the old biopolitics of eugenics, but a new biopolitics which searched for susceptibilities or predispositions in individuals, which aimed for pre-symptomatic diagnosis, that is to say, to diagnose disorders before they became evident in frank diseases, and which thought crucially in terms of risk, precaution, and prevention. And if you look at the way in which the politics of genomics is organized today, those are the key features of that new genomic biopolitics. Identify risks, identify predispositions, intervene early. Earlier is always better. Operate on the precautionary principle. Try and find the seeds of disorders before they emerge and counter them at that point. And you might want to argue, and I think this is a legitimate argument, that that idea of prevention, precaution, preclusion, prediction, preemption, which one sees not just in genomics but in politics more generally, in a whole range of areas, has become the kind of leading mantra of the role of biological understandings in disease prevention. Nonetheless, we need to recognize, and this I think is what I mean by engaging with the truth discourses of the science, that what we know about genomics is only one thing. What we know about genomics is the more we know, the more we don't know. The more we understand, the more message from up above, the more we understand, the more we realize we don't understand. I happened across this paper by Eric Lander a couple of years ago on what we knew as a result of the human genome. What we know as a result of the Human Genome Project and subsequent investigations is only about 1.5% of the human genome can be understood as coding for proteins. In other words, what we used to call genes. A 6%, further 6% must be doing something because it's evolutionary conserved, but it doesn't code for protein. There are millions of conserved elements that we don't know anything at all about. We don't know what they do, we don't know why they're there. There are thousands of other sequences that have unknown roles in things like cell cycle regulation, perhaps immune responses, perhaps brain process. And anyhow, we know that these sequences, which we used to think were so important, are really not what is important, because a sequence is in a single dimension, whereas chromosomes are folded up in all sorts of complicated ways in 3 dimensions. We're beginning to understand the roles of these little SNPs, these polymorphisms, but we don't understand the reason why in some areas of the human genome, whole chunks of the human genome are repeated again and again and again, sequences repeated many, many, many times. We no longer believe that every common disease has a genomic basis, even if it's complicated. Perhaps the most we can say is that common diseases are the end point of many different rare genomic variations. And even where we have a very clear idea of heritability, because we can trace things down family generations, the proportion which is explained by our knowledge of the gene sequences is very small. It's conventional to think that we know a lot about the genomics of breast cancer, and that we know about the heredity of breast cancer. And we do know about forms of breast cancer that are passed down from generation to generation, but the amount of that passing down of the sequence from generation to generation that can be explained by what we know about the gene sequences is very small, about 20%. The rest of it is what's conventionally called the missing heredity. So critical friendship in this respect of genetics involves, to my mind, an understanding of the science, an understanding of what the science has demonstrated, and an understanding of the limits of that science, and a repeated willingness against those who claim we know so much to say the more we know, the more we don't know. And I just want to give you a little example of this from the work that I've been doing around psychiatry. So as I said, one of the characteristics of contemporary genetic biopolitics around disease is the search for biomarkers. That is to say, the search to identify genetic sequences that will enable you to predict whether or not an individual is going to develop a disease. Those were the kinds of things that Leroy Hood was talking about in that quote that I gave you earlier on. So these calls for genetic screening, perhaps for whole genome screening, these investments in biobanks to try and link genetic data to lifestyle and medical records, all organized around the wish for early intervention, bringing the future into the present, bringing your future biological destiny into the present, and acting in the present in order to manage that future, to misquote Ian Hacking's work on risk here. Now, the logic of this kind of biopolitics seems irrefutable. Surely it is better to prevent rather than cure. Surely it's better to intervene early rather than intervening late. Surely it's better to screen populations, to identify those at risk of disorders, and to characterize those disorders, and to intervene, perhaps with surgery, the famous case of Angelina Jolie and her double mastectomy, perhaps with drugs if you think the child is going to develop antisocial behavior disorder, perhaps with some other interventions if you can identify the genetic basis of attention deficit hyperactivity disorder, or perhaps of autism, or perhaps of Alzheimer's disease. Obviously, you have to screen and intervene. Who can argue against that? But however, if you want to be a critical friend in the sense that I'm talking about, you need to begin to think about what those screen and intervene technologies might actually do. What are the costs and what are the benefits of screening? How many do you have to screen to prevent one death or one untoward incident? How many false positives? How many false negatives? Before we think about screening for psychiatric disorders, which is the area where I've been looking at most closely, perhaps we should look at some other disorders. You won't be able to see this, but on the bottom of this screen here, there's a picture of the person who invented the prostate-specific antigen test, widely used in the United States to screen men, to identify men who are at risk of developing prostate cancer. And on the basis of this indicator, the PSA indicator, to intervene, often by surgery and drugs, to remove the prostate of those men, causing them great discomfort, sexual dysfunction, and many other disorders. The picture on this slide here, which you probably can't see, is a picture of the inventor of the prostate-specific antigen test. And the quote that is on this slide is that inventor saying that this was America's greatest public health disaster. Greatest public health disaster because it resulted in thousands of interventions into the lives of men who would never have developed this disorder at all, or if they did develop prostate cancer, would die with prostate cancer rather than dying from prostate cancer. Breast screening is perhaps an even more controversial example. When I first talked about this problem in California, amongst an audience which was largely female, I was practically run out of town for the suggestion that screening asymptomatic women for breast cancer and intervening on the basis of those screenings was perhaps not an unqualified good. This is a quote from one of the Cochrane reviews, a meta-analysis of screening for breast cancer. And this is what they say: screening led to 30% overdiagnosis or overtreatment, or an absolute risk increase of 0.5%. This means that for every 2,000 women invited for screening throughout 10 years, 1 will have her life prolonged. 10 healthy women who would not have been diagnosed if there'd not been screening, 10 healthy women will be treated unnecessarily. More than 200 women will experience important psychological distress for many months because of false positive findings. It's thus not clear whether screening does more harm than good. If you look at what's going on in psychiatry today, you will see a huge increase in the wish to find biomarkers. This is just from a little piece of research which Joel Abi-Rashed and I did in the course of our work for that book, Neuro. And that just illustrates the increase in the research papers seeking biomarkers for psychiatric disorders, often genetic biomarkers for psychiatric disorders. The search for biomarkers is perhaps the most powerful logic for guiding psychiatric research, and has been over the last 20 years. And you can see here just some of the areas where biomarkers have been sought. Biomarkers argue to trump behavior. Let's move away from symptomatic diagnoses to diagnose on the basis of genetics or on the basis of neurobiological biomarkers. But many books on the role of biomarkers, looking for biomarkers early and especially looking for biomarkers late. You won't have to go many weeks in reading your newspapers before you read of the discovery of a new biomarker that's going to predict whether or not someone will develop Alzheimer's disease, and indeed some tests that people can do when they're at home. You will see here attempts to find biomarkers for criminal traits, biomarkers for in the adolescent brain to determine whether or not they're going to develop behavior disorders, biomarkers in almost every area that you think about. So for 20 or 30 years, the logic of psychiatric research has been very heavily focused on trying to identify the biomarkers for psychiatric disorders, based in the belief that a psychiatric disorder is at root underpinned by an identifiable biological basis, whether that's genetic or whether that's neurobiological. Some of you may be aware that the 5th edition of the Diagnostic and Statistical Manual of Psychiatric Disorders was published about a year ago. That manual, which is used very widely to identify what disorder an individual might be suffering from, had always been thought, those diagnostic categories had always been thought ultimately to have a biological basis. For 20 or 30 years, hundreds of millions of dollars, hundreds of thousands of person hours have been directed towards trying to find these biomarkers for psychiatric disorders. And the hope was that by the time this new edition of the Diagnostic and Statistical Manual would be published, that it would be possible to find, quote, objective biomarkers of psychiatric disorders. When, in June of last year, that manual was published, it became clear that there was not a single clinically useful biological biomarker for any psychiatric disorder. No way in which you could find a reliable genetic or neurobiological marker that would indicate whether a person would develop a disease or would help you diagnose the disorder when you saw the individual with psychiatric distress in your clinic. Not a single one. Why should that be? Why should it be that after investing so much time, so much energy, and so much belief in the idea that there was a biological basis for psychiatric disorder, it had not proved possible to demonstrate it in any single case? Was this a technical question? Was that a problem of evidence? Did we need more research? Or was it a fundamental conceptual question about the nature of the disorder itself? So I think that is one really rather important place for critical friendship. For critical friendship, because the logic of screen and intervene, the logic to intervene early, to prevent, predict, preclude, and preempt disorder, shaped a multitude of psychiatric practices, and indeed a whole lot of other practices. What can we learn if we adopt this relationship of critical friendship from that failure? Well, perhaps we can learn something rather fundamental. It's not just that the search for the origin of diseases in genetics is more difficult than some had hoped, but perhaps, just perhaps, it's because we're looking in the wrong place. Perhaps, just perhaps, it's necessary to insist with our friends the psychiatrists and our friends the neuroscientists, to insist that an ailment, a psychiatric ailment, a period of mental distress, is inextricably an experience of a human being who's lived a life as in a social and cultural milieu. That milieu is not just an epiphenomenon, that milieu is actually constitutive of the disorder itself. That if you want to have a biomedicine that starts from the human, you have to understand what the human is like. And perhaps you have to realize that the failure of this type of biomedicine, this still old type of biomedicine that believes it's possible to separate out the biological and the social, actually hangs on a really fundamental misunderstanding of what an ailment is. Perhaps not just a psychiatric ailment, perhaps any other ailment as well. Because we know that what you've proved in the laboratory, you find very hard to prove in the clinic. Why can you not move from the laboratory to the clinic? Why has translation failed in almost every area of biomedicine? Well, perhaps the relationship that I'm calling critical friendship might make a difference. I want to give you two other rather shorter, much shorter examples before trying to bring this back to the question of what politics of life might look like in the 21st century. So the first example, or rather the second example, is that of epigenetics. As I've already said, here epigenetics is a rather contested term, but here I just want to take epigenetics to look at the way an organism's genome doesn't merely express itself during development, but is modified from conception onwards as a result of the interaction between that organism and its environment, the activation and the deactivation of genes. So epigenetics traces the way in which gene sequences are activated or deactivated across a lifetime. Perhaps the most interesting of the researchers in epigenetics is a Canadian lab, the lab of Michael Meany. And this lab of Michael Meany, which works largely on rodents, has shown that the way in which a mother rodent treats her pups, treats her baby rodent, shapes the gene expression in that baby rodent's brain, which shapes the rodent's behavior as it grows up, which shapes that rodent's relationship to its own pups, which shapes their pups' expression of their genes within the pup's brain, which shapes the next generation of pups' behavior, and so on and so on and so on. Here's a mechanism, an epigenetic mechanism for the passage of these experiences down the generations. And Meany has gone on to argue that this epigenetic kind of experimentation done with rodents also has implications for humans, that perhaps human experience from the moment of conception onwards shapes the expression of their genes, shapes the way in which those genes are expressed in the brain, shapes the way in which those gene expressions affect the behavior of the individual themselves, shapes the way in which they respond to their environment, and does so down all the generations. And very rapidly, Mike Meany and his colleagues have begun to argue that you can explain all sorts of social ills on the basis of these epigenetic arguments. So how should those of us who want this relationship of critical friendship respond? For some, epigenetics is a savior for the human sciences, because it demonstrates that what is crucial is not the inherited genetic complement that an individual is born with, but what happens to that individual over the course of their lifetime. Perhaps, they say, this is a recognition of the inseparability of the milieu and the biology. Perhaps, indeed, it could give a crucial role for the social and human sciences in accounting for the shaping of vitality at this molecular level. And perhaps it shows that the brain, no less than the psyche, is, because we always thought that the psyche, the psychological, was open to these environmental influences, perhaps it shows that the brain itself is shaped by environmental inputs at the level of the genome. So how should we respond to these experiments which are based on rodents? Should we react with horror to the fact that humans might share something with rodents? Or should we work with those researchers? Should we try and move them beyond a kind of reductionism? Should we move them beyond a rapid extrapolation of their work into the human realm? Should we try and understand a little bit more what these environmental inputs might be? And should we, on that basis, collaborate, bringing sociology once more into a kind of alignment with what the life sciences are telling us about the nature of human beings? Well, as I said right at the beginning, if you're going to take this seriously, this means engaging in some rather dangerous and risky collaborations. And that's what my group at King's is doing in relation to the urban brain in our, what we call our urban brain lab. We've known for 100 years that the pattern of distribution of psychiatric disorders across urban space is not uniform. We've known for 100 years that there are certain concentrations of psychiatric disorders in particular regions of the city, in inner regions, in regions of poverty, in regions of high density, in regions of social deprivation, in regions of racism, and regions of exclusion. Social epidemiology and social psychiatry has traced that out again and again and again over 100 years. Are we content with just describing those distributions, or might it not be relevant for our sciences, for the social and human sciences, to try and understand something about the mechanism, about the way in which the experience of the urban gets under the skin and shapes body and brain, the way in which social suffering is not just something external to the corporeality of the human, but something inscribed within their very bodies and minds themselves. That's not a particularly new argument. If you look at some of the earliest work in sociology, if you look at the work of the Chicago School, you will see that the Chicago School refused that distinction between the biological and the social that became the common sense of the social sciences in the second half of the 20th century. This is Robert Park and Burgess writing in 1925. The city is not merely a physical mechanism and an artificial construction. It's involved in the vital processes of the people who compose it. It's a product of nature. And there were many others within the Chicago School who sought a kind of vitalistic sociology to understand how the experience of living in particular urban environments shaped the bodies and the souls of those who lived in those places, and who felt that this kind of vibrant, vital way of understanding the social experience of individuals was absolutely crucial if you were going to understand that distribution of mental disorders that the epidemiologists had shown us again and again and again. Now, in the almost 100 years that passed between those early sociological investigations of mental life in the metropolis, and those investigations go right back to Georges Simmel and his famous paper, Mental Life in the Metropolis. They go back to Walter Benjamin and his studies of Berlin and his studies of Paris. They go back to Charles Baudelaire and his idea of modernity. In the late 19th and early 20th century, the idea that there was no distinction between a social explanation and an explanation which looked at the vital processes of the human, that indistinction was taken for granted. Now, something happened in the second half of the 20th century which made that collaboration pretty much impossible. And if you look for those who are doing that collaboration now, you don't find them initially amongst the social scientists. Oddly, you find them initially amongst the neuroscientists. This on the board here, that's a picture of a German neuroscientist called Andreas Meyer-Lindenberg, whose work gained a certain amount of attention because of his argument that you could map social stress in the brain of urban inhabitants. That if you did brain scans of those who'd been brought up in the city and those who'd been brought up in the countryside, you saw that they had radically different ways of responding to stress. That something about being brought up in an urban environment was actually inscribed within the brain itself, and that throughout that individual's life, it led to them responding differently to stressful environments. And that you could trace that out by looking at patterns of activation in the amygdala when an individual was exposed to something called stress. Now, we all know what stress is, perhaps, or perhaps we don't know what stress is. Stress has become an all-purpose mediator between the objective and the subjective. Stress appears to be an objective phenomenon. You can map stress perhaps across urban environments, and you can begin to see how stress gets into the brain. But what is stress? Is everything that those of us who've worked on the sociology, on the anthropology, on the investigation of urban life, is all that to be reduced to one single mediating term, stress, or is there any way that we can get beyond this? This is the inventor of modern stress theory, Hans Selye, incidentally, as everybody says, funded for most of his life by the tobacco industry. Hans Selye says, if you can read this quote, everybody knows what stress is, and nobody knows what stress is. So perhaps, just perhaps, if one is trying to think about another area of critical friendship between the social sciences and the life sciences, one of the things that one might usefully try and do is to break down this idea of stress, to try and explore how the experience of living in the city, how the understanding of the city, how the culture and history and mental life of the city itself shapes the way in which these things get inside the brain. Perhaps we can begin to make a link between how people live on the street, between their experience of racism, their experience of exclusion, their experience of discrimination, and the way in which these forms of social suffering get inscribed into the body and soul themselves, because they do indeed, and they lead to radical shortening of those people's lives with dire consequences. If you look at all these arguments about stress, though, what they refuse to look at is precisely those things that the social and human sciences focus on, which is to say mental life itself. How an individual codes their experience, how they give meaning to their experience, how they make sense of that experience. Surely that's the kind of thing which us in the social sciences might try and contribute to this kind of analysis. And if we wanted a justification for that, we need to go no further than to look at the inventor of social neurosciences, John Cacioppo. John Cacioppo has got a good justification to be the person who invented the term social neuroscience, has done some intriguing work on loneliness. Loneliness, says Cacioppo, loneliness can kill. People who are lonely die younger. But loneliness, and this is the intriguing thing about his research, loneliness for Cacioppo, that which kills, is not the quantitative number of social contacts that an individual has, it's their sense of social isolation, their belief that they are isolated, their feeling that they are cut off from their fellow human beings. It's the subjective sense of isolation, shaped by beliefs about culture, shaped by history, shaped by all those things that we have studied so long in our social and ethnographic studies of urban life. It's those beliefs that determine whether or not an individual shall suffer the consequences of loneliness, not some objective factor. So as soon as you begin saying that it is the subjective sense of social isolation that gets under the skin, it seems to me you open up an intriguing place for critical friendship. Given that over half the world's population now lives in cities, and given that the experience of social isolation in cities, if our ethnographers are to be believed, is growing intensely as cities get bigger and bigger and bigger, that seems to me to be not an unworthy area for critical friendship. Okay, I'm going to draw this talk towards a close now. I was going to say a little bit about synthetic biology, which is an area where I've worked. Synthetic biologists probably have a relationship to life which is the most mechanistic that you can possibly imagine. Synthetic biology tries to break living processes down into a series of small, definable parts, genetic sequences, and then engineer and re-engineer organisms by modulating those genetic sequences. And indeed argues that once you begin to understand that process, once you begin to understand that you can move from reading the genome to writing the genome to actually creating these genetic sequences out of DNA bases that you make from sugar, then finally you have reached the end of vitalism, because life itself can be created from inorganic processes. But I think if you begin to understand, again, what's known and what's not known, you realize that a great deal of this is great overstatement. For instance, and this is an interesting quote from a synthetic biologist themselves, which again, as with the genome, just indicates how little we know about these vital processes. This is Rob Carlson writing in a book called Biology is Technology. He says, a Boeing 747 consists of about 50,000 kinds of parts, some 6 million total components, and we know the precise specification of each of those parts. But a relatively simple single cell, such as yeast, has got millions of moving parts, most of which are unknown, approximately 6,300 kinds of genetic parts, of which we can name about half, but for most of which we have no design specification at all. A human body has something around 10 to the 14 or 100 billion cells, not to mention all the microbes that inhabit it, and the human brain contains about 100 billion neurons, each of which are different, with 100 trillion synapses of connections between them. So the amount that we do not know about human biology is immense. The more we know, the more we don't know. And the lesson for critical friendship then is to keep insisting on that constant reminder that we know so little about human or animal vital processes. And indeed, the thing that we don't know about vital processes is their vitality itself. Because although we can characterize much of human biology at the molecular level, what we are unable to understand at that molecular level is the complexity of the dynamic, self-organizing, emergent characteristics of living creatures that exist in space, in time, and their milieu. So here, I think, the role of the social and human sciences is partly to insist again and again and again about how little we know and how dangerous scientific hubris might be, but also to engage with those life scientists, to engage with them specifically around the question of the human, if their claim is to be a genuinely human science. All right, a few words in conclusion then. I mentioned at the beginning the work of Didier Fassin and his suggestion that another politics of life was possible. And I suppose what I've tried to do in a rather inchoate way in this talk is to suggest something about what that other politics of life might be. Fassin has this conception of life as such. Life, he says, as the course of events which occur from birth to death, which can be shortened by political or structural violence, which can be prolonged by health or social policies, which gives place to cultural interpretations and moral decisions, which may be told or written. Life which is lived through a body, not only through cells, and as a society, not only as a species. Life as such. And I suppose what I've tried to suggest, as I say, in this inchoate way, is how a politics that engages with our understanding of life as such might gain some traction on the enormous power that the life sciences are having in our contemporary world. And I want to try and insist, and that's probably why I've bored you with all the details of these little examples here, that the way in which one needs to do this is to try and engage meticulously with the truth claims of the scientists themselves, with the operative philosophies, the way in which they actually work, the way in which they actually seek to experiment and frame their explanations, to relate to those with what I've called critical friendships, not just to trace out the historical conditions of these truth claims, not just to trace out the rules of the game. We've become quite good at tracing out the genealogical and epistemological and ontological rules of those games, but to try and grasp what's at stake with those games, to go beyond genealogy, to go beyond critique, and try, as I've said, to make the life sciences genuinely human sciences, to understand the way in which human vitality itself is constrained by social injustice and inequality. My favorite historical epistemologist, Georges Canguilhem, in his wonderfully elliptical way, has a couple of phrases that I've found suggestive. He says, "The thought of the living must take from the living the idea of the living." He says, "Every mode of biological reason is in a certain way also a philosophy of life." So our sense of ourselves as living human beings, our ethics, our self-technologies have become inextricably tangled with the truth claims of the contemporary biosciences. And because they become so entangled, I think we're obliged to try and find a new relationship with those, to move beyond description, beyond commentary, beyond critique, to have a kind of affirmative relationship to these truths, an affirmative relationship to the arguments for a non-reductionist biology of organisms, to bring these arguments into conversation with the social and human sciences, but at the same time to recognize that our social and human sciences are also sciences of the living. And I think if we did this, those of us who work, especially in my discipline, sociology, can't fail to be aware that it's held in rather low esteem by those who govern us. It's not looked to find arguments of our contemporary plight. Sociological experts are not on the radio. Sociological experts are not advising government. Sociological experts are not analysing the new forms of injustice that are emerging in the world. Sociology appears to have lost its traction. And I just want to suggest to you that perhaps, just perhaps, this new relationship between the social and the life sciences that I'm advocating might help regain that traction and regain a capacity of those disciplines to remake our human world for the better. Now, of course, a talk wouldn't be complete without a reference to Michel Foucault. And you will recall what Michel Foucault says at the end of the order of things. At the end of the order of things, he suggests the possibility that development within philosophy might be transforming our understanding of the nature of the human. And what I want to suggest to you right now is that perhaps it's not to philosophy that we look to transform our conception of the nature of the human, but perhaps now we should be looking to the life sciences. So this is what Foucault says: amongst all the mutations that have affected the knowledge of things and their order, only one, which began a century and a half ago and is now perhaps drawing to a close, has made it possible for the figure of man to appear. That humanist figure which set the human apart from the animal, apart from its world, and gave us our belief that we had a special place within nature, and man was the explanation and the center and the key organizing principle of our knowledge. And Foucault says, if those arrangements were to disappear, if some event of which we can at the moment do no more than sense the possibility were to cause them to crumble, as the ground of classical thought did at the end of the 18th century, then one can certainly wager that man would be erased like a face drawn in sand at the edge of the sea. Well, perhaps it's not to structuralism or to philosophy that we might look for that event of which we can at the moment do no more than sense the possibility, but perhaps we should look to the life sciences themselves. And I'll stop there and thank you for your attention.